For decades, the clinical picture of Alzheimer's disease has centered on the cerebral cortex and the accumulation of amyloid plaques in late life. The very first abnormal protein deposit anywhere in the human brain appears in the locus coeruleus. This tiny brainstem nucleus develops hyperphosphorylated, truncated tau decades before amyloid reaches the cortex. This timeline chart highlights the massive temporal discrepancy between the two pathologies. While cortical plaques typically manifest after age 70, the first abnormal tau in the locus coeruleus is detectable in the majority of people from age 25. Previous analyses trace how this pathology burns across the cortex during the disease's middle passage. Those accounts assume a starting point they do not describe. We are going to strictly derive the chemical ignition of that initial pathology. By understanding how one specific localized nucleus poisons itself, we locate a phase of the disease that is entirely cell autonomous, the only window where the chemistry is contained enough to be extinguished. To understand the ignition, we have to look at the tau protein itself. In a healthy cell, tau's job is to bind and stabilize the neuron's internal transit tracts, the microtubules. Its grip on those tracts is controlled entirely by its phosphorylation state, how many chemical phosphate groups are attached to it. That phosphorylation is a dynamic equilibrium. It's written by enzymes called kinases and erased by opposing phosphatases. This balancing scale visualizes the equilibrium. On the left is the writer, kinase GSK3-beta, constantly adding phosphate to tau. On the right is the eraser, phosphatase PP2A, which removes them. If one side overpowers the other, tau's shape alters, detaching from the microtubule. A persistent shift in this balance is the mechanical basis of the pre-tangle. In the locus coeruleus, that shift is driven by a confluence of structural and chemical failures. The locus coeruleus is unusually exposed. A single neuron in this nucleus projects a massive, diffuse, unmyelinated arbor that reaches across vast territories of the forebrain. Most critical neurons defend themselves with a perineuronal net, a condensed structural matrix of sulfated sugars that acts as a physical shield. Neurons wrapped in this net strongly resist tau accumulation. This schematic of a healthy cell membrane shows one reason why. The protective net provides a specific surface of N-sulfated heparin sulfate. The protective protein reelin requires that exact surface to properly stage its receptors. When this interlocking three-part mechanism assembles, it sends a continuous signal to suppress the writer kinase. The locus coeruleus constitutionally lacks an aggrecan-based perineuronal net. Without that sulfated staging surface, the reelin receptor lock physically fails to assemble. Because the break cannot be staged, the suppression signal never arrives. In the locus coeruleus, the primary tau writer runs under-restrained from birth. This nucleus does not fall victim to tau by random chance. Its native architectural makeup leaves it defenseless against the very enzyme that begins the disease. Beyond its anatomy, the locus coeruleus is defined by what it manufactures, the catecholamine neurotransmitter, norepinephrine. Catecholamines are necessary for communication, but highly dangerous if left loose in the cytosol. A healthy cell manages this hazard by pumping norepinephrine safely inside synaptic vesicles, using a transporter called VMAT2. This intracellular diagram demonstrates what happens in the presence of ApoE4, the primary genetic risk factor for the disease. The ApoE4 protein physically binds to the VMAT2 pump, creating a blockade that stops the transporter from functioning. This genetic blockade is compounded by the neuron's activity. Chronic stress and high firing rates cause the cell to rapidly pull neurotransmitters back in, overwhelming the remaining uptake capacity. With the vesicle blocked and reuptake overloaded, sequestration fails. Norepinephrine escapes containment, flooding the surrounding cytosol.
Waiting in that cytosol is monoamine oxidase A, the enzyme responsible for degrading loose catecholamines. As the enzyme processes the escaped norepinephrine, it produces a highly reactive, toxic aldehyde known as DOPEGAL. Because only norepinephrine neurons utilize and metabolize norepinephrine, this specific chemical cascade cannot happen anywhere else. DOPEGAL is produced exclusively in the locus coeruleus. The selectivity of the first lesion is a consequence of this unique chemistry. The locus coeruleus is the only population of neurons in the brain that manufactures the metabolic trigger for the ignition. DOPEGAL acts as a targeted biochemical activator. It rouses a specific enzyme from dormancy. Its target is a dormant lysosomal cysteine protease called asparagine endopeptidase, or AEP. Here we see that interaction. DOPEGAL molecules lock perfectly into the dormant structure, triggering AEP to unfold into a highly active state inside the neuron. Once active, AEP commits its first crime. It targets the tau protein and cleaves it directly at the asparagine 368 residue. This structural cut is far worse than basic phosphorylation. The truncation outright destroys tau's ability to grip the microtubule, leaving behind a severed fragment that is primed to aggregate into a self-templating seed. At the exact same time, the identical AEP enzyme commits a second crime. It targets a completely different protein in the cell nucleus, known as SET, severing it into fragments. These severed SET fragments do not degrade. Instead, they retain a concentrated inhibitory power and rapidly translocate out of the nucleus and into the cytoplasm. In the cytoplasm, the fragments seek out the PP2A eraser. They physically bind to the phosphatase and completely silence its ability to remove phosphate from tau. Looking back at our scale, we see the systemic collapse. The writer runs without its break. The tau protein itself is severed into an aggregation seed, and the eraser is permanently gagged. Clinical interventions struggle to clear the pre-Tangle tau because they face a locked system. A single metabolic switch simultaneously manufactures the toxic seed while disarming the cell's only biological method of clearing it. Zooming out from the intracellular chemistry, we have to consider the anatomy of the Locus Coeruleus again. The N368 tau seed created by that cut is highly prone to propagation. The physical consequence is massive. The diffuse arbor of the Locus Coeruleus perfectly positions it to broadcast these transmissible tau seeds throughout the forebrain. As the subcortical neuron exports its damaged proteins, recipient neurons take up the seeds. This progressive broadcast triggers the massive, self-sustaining loops of cortical tau and amyloid that characterize the later stages of the disease. This sequence dictates a strict therapeutic corollary. Billions of dollars spent attempting to clear the late-stage cortical fires have failed because a conflagration no longer requires its original spark to burn. The pathology is structurally vulnerable to intervention only when it is cell-autonomous, confined to a single nucleus, and entirely dependent on its own self-poisoning chemistry. This mapped layout identifies 3 precise, druggable targets before the broadcast occurs. We can disarm the AEP protease directly. We can starve the enzyme by inhibiting MAOA to stop DOPEGAL production, or we can cut off the chemical leak at the source by restoring the VMAT2 pump. To be clear on the scientific ledger, DOPEGAL production, AEP activation, and the exact N368 tau cut are directly measured empirical findings. The absence of the renin staging surface is a rigorous structural inference derived from the established lack of a perineuronal net. Alzheimer's disease ignites decades early in a small brainstem nucleus, poisoning itself with the precise chemical required to maintain our daily vigilance. If we map the chemistry of the Ember, we possess the only mechanistic target small enough to extinguish the fire.